this is the stuff that you guys need to know for the next quiz. We're gonna try and get all the way through antifungals, but if we can't get through antifungals, no big deal. So what we're also going to do is then we are going to take a break. Let me turn this on. Then we are going to take a break and I thought maybe we could just come back to this classroom instead of sitting in the lab and doing our review in the lab since it's comfortable and, but we'll take a break in between. There you go. Okay, so we'll take a break and then we'll come back and do our lab review. Okay, so last time we talked about fungal organisms, we talked what fungi are, we talked about morphology between yeasts. I can't move this up because then it's just going to confuse you because we have all our lab stuff under there. We talked about yeasts versus molds versus dimorphic fungi. Oh, really quick, I'm gonna do one of my little spiels. Everybody ready? Okay, it's gonna be a spiel. So, okay. My analogies are really funny, just so you know how my brain works. So, I went to a meeting in my daughter's school, and they gave us a meeting about how to talk to our kids about sex and like, okay, what do we need to do? Like, how do we prepare? They're starting to teach repro and like, okay, what are we teaching in repro? How do we talk to them about consent and dynamics of relationships and all that kind of stuff? So what she started off telling a story about Sleeping Beauty. And so they read the story of Sleeping Beauty. And the reason they read the story is because the parents of their daughter, she was cursed. If you guys know the story of Sleeping Beauty, she was like cursed, and they said she's gonna prick her finger on a spindle and then she'll die. But then they changed it to where she'd just fall asleep. So what the parents did is went through the entire kingdom and got rid of all the spindles, right? They burned all the spindles to the ground. So her point in telling us this when she's talking about reproduction is she's saying, don't burn all the spindles to the ground. Teach your kids how not to touch a spindle, what a spindle is, how to protect yourself, and this is right, and this is wrong, and this is good, this is bad, this is, and so don't go through and try and protect your kid from everything because it's not gonna work out, it's just not feasible. So this relates to you. How? Because I noticed that in class, some of you aren't paying attention. So some of you are on Instagram, some of you are doing your pre-labs, some of you are reading and doing other things. So I don't want to say I don't care because I really care. I really care about you a lot, and I really want you to succeed, and I really want you to be able to do all the things and to be able to get everything you want. But should I make all the rules to make you study and make all the rules to, okay, well, pre-labs are due at the beginning of class then. Nobody can do pre-labs during class because I want you to pay attention because I want you to succeed. Well, I feel like to me, it's almost like I'm destroying all the spindles so that you don't, it has to come from you. And I can hopefully help teach you how to not be distracted by those things, how to focus in class, how to study best. I will try and teach you those things, but also you do have to be here. And this is something I have learned throughout my years of going to college. Some days I would have it, some days I wouldn't. Like, I would go to organic chemistry and be like, ugh, and like, my head couldn't stay vertical. And I was like, why is this so hard to stay awake? So I would give myself tools. So I'd bring 4 different kinds of gum. I'd bring lollipops. I'd bring popcorn. I'd bring a bubbly water. I'd get up and go to the bathroom every 20 minutes or something if I had to keep myself awake and keep myself going. And for me, I have to write. I have to physically write to be present. And I draw pictures and I draw arrows and whatever it is. If you do that on your iPad, 100%, but just the physicality of doing something while you're listening helped me. So I don't want to make labs due at the beginning of class because that. I don't want to babysit you. I don't want to make you do something that you don't want to do. I don't want you to do something you don't want to do. I want you to want to do it, and I want you to be here. So that's my little spiel for the day. But one thing I do want to make sure is that I am helping you if you need help, because that is a real thing. The people who use tutors are always the better students. The people who get the extra credit and use the extra credit are always the better students. It's never the students you want to use it. I want everybody to use it, but it's never the students who really need it. So do the extra credit. Ask me if you need one-on-one. I'm happy to do one-on-one with any of you. And I know this was another thing that we've been talking about. I'm taking this like an equity research class through the college of like equity research through the whole district and like our Folsom Lake College, and how are we doing as a college, how am I doing as a professor to like reach all of my students equally, depending on some foster students or socioeconomic backgrounds or cultural backgrounds. Like what am I doing individually and how can I improve? And then how can we improve as a college? And one thing that stuck out to me is because I always try and like, okay, reach out to every student who needs help regardless of what their backgrounds are. But there's two parts to that, is that one part, I don't know what you have going on in your life. And sometimes my life is totally nuts, but I leave that at home. I try to be here and try to like, okay, leave that home, be here as much as I can. But I know for students that sometimes can be really hard. It can be really challenging to leave your stuff at home and to be here in class. So if you need just an ear, if you need somebody to talk to, I would like to be that person for you. But if you don't, I will put you in touch with somebody. So reach out to me. But the second thing I wanted to say about that is that, I try to be approachable, but I understand some people don't want to approach me just because of our dynamic. Like, I hope I don't like make myself to where I'm here and you guys are here, because I want to be here with you guys, and I don't want to be like I'm on a different level than you. So I want to be able to converse as if we're colleagues, as if we're friends, and then have a dynamic where you can talk to me. But if you don't feel comfortable talking to me, I completely understand as well. So if you want to just shoot me a little email about something that's going on in your life, shoot me a little email about how I can help you better, or just I need a little assistance right now, then I can just put you in touch with our success coach. Our success coach is a great person to talk to. He can help with all kinds of things. So if you don't want to talk to me, I totally understand that as well, but I don't know how to help you if I don't know that you need help. So all of that being said, just know that I'm here. We have a group of students that are excelling, that seems like, I know the material's not easy, but it seems like they're thriving, and but then it seems like we have a group also that's not thriving. So I want to make sure that I help everyone, and then just reach out to me how I can help you, or if I can put you in touch with somebody that you're more comfortable receiving help from. But either way, being present in class, 100%. You have to be here anyway. If you don't study here, it's twice as much time when you're alone, when you'd rather be doing something else. So when you're here, like, try to get your brain to like 100% be here. Okay, that's my rant for the day. All right, so we talked all about yeasts and molds and what those look like, the morphology of yeasts and molds, some of the unique structures that make fungi unique, such as manoproteins, glucans, and chitin. We talked about their internal structure is very similar to our internal cell structure, except one cell of a yeast can go live on its own where our cells can't. We talked about what they might look like under the microscope. We talked about mycelium and hyphae. We talked about the Spitzenkorper, about septate and non-septate fungi, that they form these septums. Septum is probably just the, septa is probably the plural actually. We talked about ergosterol is the cholesterol in the cytoplasmic membrane of fungi. aerial versus vegetative hyphae. We talked a tiny bit about spores. We'll talk more about spores today. So we talked about sporangiospores and conidiospores. What else? Dimorphic fungi is generally based on temperature, and that was it. So today we're going to talk about fungal metabolism. So fungal metabolism, the reason I kind of like to organize the class the way that I do, where I talk about bacteria and then everything that has to do with bacteria. Now that we get to fungi, then we can see a little bit how they're unique, but then also it's a good review. So now we can do our metabolic review. What is an aerobic organism? What is an anaerobic organism? What is a facultative anaerobe? All that kind of stuff. Okay, so yeasts are facultative anaerobes. It's nice. We don't have 20 different kinds of, they're all doing different things. Yeasts are facultative anaerobes. Molds are strict aerobes. So what does this tell us about the organism? Like when we're talking about clinical medicine, what would this tell us about the organism? Like,
where it can thrive.
Yeah, like where, maybe like it's tissue tropism, right? Like where that organism will exist in the human body as a pathogen. Where will that organism cause infection based on its access to oxygen? So yeasts can be anywhere, right? And then molds have to be either in the lungs or on the skin. We have some little examples of molds, how they can kind of grow other places and they can kind of set up shop other places, but I'll show you why and what that means. Okay, so aerobic respiration, remember aerobic respiration there's 4 stages, so let's do them all together. The first stage of aerobic respiration starts with glycolysis. So glycolysis, we have glucose that turns into pyruvates. Okay, so then the next step is called the intermediary step, and that step goes 2 pyruvates to acetyl-CoAs. So now we have 2 acetyl-CoAs. Now we're moving into the third step, which is Krebs cycle, so that you get 2 acetyl-CoAs from the Krebs cycle, and the whole goal of the Krebs cycle to get out of it is what? Yes, NADH and FADH2 is our whole goal, are those hydrogens attached to those little taxi cabs. We want those hydrogens. So now in the fourth step is electron transport chain. We take those hydrogens, pump them across a membrane. Hydrogens spin the ATP synthase, add a phosphate to ADP, and we have a ton of ATP because that oxygen is helping pull the electrons and pull those hydrogens down to make water. So oxygen is our final electron acceptor. I know we kind of forget that because it doesn't form our final product, which is ATP, but oxygen is the battery that makes the whole car run. So yes, the goal of your car is to get somewhere, but you can't do it without the battery. And yeah, you need gas, but you also need a battery. And so the battery is oxygen. Don't forget that, because that's why we breathe. And in this whole process, we form CO2, and that's why we breathe out. So when we're exercising, working out hard, and we're breathing and gasping for breath, it's because our mitochondria are pumping out CO2 really rapidly, and that CO2 is wanting to leave the body in exchange for oxygen. Oxygen comes in, CO2 goes out, but that's why, is because we're making ATP. Okay, so a facultative anaerobe, so molds are aerobic, and then yeasts are facultative. So they can do all those 4 steps, or they can do glycolysis, or the intermediary step. So I have a slide to remind you what that is. Where does ATP synthesis occur in molds? I already kind of told you. Mitochondria. Awesome. So it occurs in the mitochondria of fungi, same as us. over all eukaryotes. Okay, so here's a facultative. Our yeasts are facultative, so they can do this, or they can use glucose, pyruvates, form acetyl-CoA to form ethanol, acetic acid, lactic acid. Yeah.
And this just takes place in like the cytoplasm, right?
This takes place in the cytoplasm. Yes, yes. Awesome. Great question.
In the mitochondria and the cytoplasm.
So all of glycolysis and the intermediary step happens in the cytoplasm, and then the acetyl moves into the mitochondria, and then goes through the Krebs cycle in the mitochondria. Yeah, great questions. So here, this is a great review also for lab, is that here we're producing acids. This one is our neutral end product. This is our neutral end product that is when we test for MRVP. I'll bring this up again when we do lab review, but it's we're looking for acids versus a neutral end product. But either way, these are products of fermentation. And so I've said this once before, but hopefully it stuck a little bit, is that anaerobic respiration is not as efficient. It doesn't make as many ATPs. And it doesn't make as many ATPs, and then it has this byproduct left over. We can't break all of those carbons apart. We can't use all of those carbons efficiently. So you end up with some sort of leftover carbon material, and that leftover carbon material is usually the acid or neutral byproduct. So we have leftovers. It's less efficient. We can't really use everything to be able to make ATP. Okay. So ATP synthesis will occur. It'll occur in the cytoplasm and the mitochondria. Okay, so metabolism, I'm going to talk more about decomposers and what they do. I'm going to talk about their ecology and where they live. I'll talk about that today. Byproducts, we already talked about that. So you can read this on your own, but let's go through, I have slides on it. So here it'll talk about aerobic versus facultative, anaerobes, and so read through those. So just one little note that I wanted to say about fungal byproducts is that we use them for all kinds of stuff. We use fungal byproducts for sake, soy sauce, tempeh. We use them for beer, wine. Those are just the ones off the top of my head. We use them we can use them for herbicides and pesticides. We use them for, I don't know, any millions of things. We use them for antibiotics, right? Our one antibiotic example that we talked about is a byproduct of fungal metabolism in some way. So they are producing those beta-lactams. Those beta-lactams are what is an antibiotic. Okay. So here we are talking about again commensalism versus mutualism. I hate being stuck over here, but my battery is dead. So commensalism versus mutualism versus a pathogenic organism or parasitism. So we do have a lot of fungal organisms that live on us we're not really sure in some cases if they are beneficial or if they're non-beneficial. And some cases we know they're beneficial, but they just live on and about us. So we do have mutualistic organisms on us that are fungal. We have commensal organisms on us that are fungal. And then we have parasitic fungi, which are called mycoses, are diseases that are caused by fungus. So I'll go through those. So when we are talking about fungal ecology, this is kind of a new branch of what we've kind of been talking about. They are parasitic, but also they're something called saprobic, or they're called saprobes, and that means that they digest decomposing material. So if you think about back in dinosaur Jurassic period, back in the day, when any log, tree, soil, animal, anything is decomposing, it is decomposed by bacteria, but mostly it's fungi. So mostly they're fungus or fungal organisms that will break up the carbons, and so that we can use those, and that we can use them, and that soil can use them, that plants can use them. So really, there are main organisms that decompose everything in our environment. So without them, we wouldn't have gasoline. Our gasoline comes from decomposed material. And so that decomposed material, millions of years ago, all came from carbon from trees and plants and animals, and that were decomposed by fungal organisms. So those are called saprobes. They're generally, I wouldn't want to say all, but mostly considered saprobic organisms. Most of them are. So they're hugely beneficial in the carbon cycle. Obviously, we need those carbons back. So when carbons are broken down by fungal organisms, then other organisms can use them. So yeah, it was just a little cartoon. I kind of liked to look through this a little bit and like, okay, where does carbon go? Then where does carbon go? So we need those organisms that are saprobic to break up those carbons. So we do also have beneficial fungi that live in and around us, and that's called our mycobiome. So we said our microbiome, sometimes it's called mycobiome. So myco is another prefix for fungal organisms. Sometimes I've heard it called the fungome, as some people say that, but the mycobiome people say as well, and that's just the organisms that live on and around us that are commensal or mutualistic. Yeah.
Would microbiome be more directly related to bacteria, whereas microbiome?
Yeah, microbiome is kind of like a broad term for everything that lives on us, but mycobiome is kind of a newer term where they're kind of gearing, like looking more at fungal organisms specifically. But I won't ask you that, just broadly, so that you know, like microbiome, they're kind of talking about everything, but mycobiome, they're talking about specifically about fungi. So we do have a lot of different, these are the genus that live on and around us. So here's Candida, Saccharomyces, Ascomycota. We have all kinds of different, here's Aspergillus that can cause that is black mold. Penicillium we have on and around us all the time. So remember I showed you the video of the spores that are in the air? There are fungal spores everywhere. And if you tried to put some strawberries on the counter, you can see how long they would last without getting fungal fuzz on them. Yeah?
We have commensal penicillium. Do we have like a slight natural antibiotic built in?
That is a good question. I wouldn't know if it would be enough, like it would be concentrated enough. Yeah, that's a good question. I'm not 100% sure. Yeah, good question. So we do, there are fungal organisms everywhere, just like bacteria. There's bacteria everywhere. But this is going to come into play when we talk about diagnostics. So just kind of keep this in mind when we talk about fungal diagnostics, about how to diagnose your patient if they have a fungal infection. Okay, so let's talk about cytopathic effects. So remember we talked about cytopathic effects of bacteria. What were our cytopathic effects of bacteria? Yes, endotoxins and exotoxins. Awesome. Endotoxins and exotoxins. So those are how bacteria will kill our cells. So how bacteria affect our cells. So let's talk about how fungal organisms affect our cells. So they can affect our cells through direct invasion. So direct invasion happens because of there's the structure that's on a fungal hyphae, for example. So if there's a mold, or sometimes remember it's called a filamentous fungi, you have a mold, and that patient has a mold infection in their lungs. What is the structure? This is just from Tuesday, so it's no big deal if you can't remember. That is, we worry about, that's dangerous because the hyphae can bury into the tissue. It's not a funny name. Spitzenkorper. Yes. So they can, so molds can dig straight into the tissue. So they're just, you know, like, if you can say it's cytopathic, but basically what it's doing is just digging, and you don't want anything digging through your tissues, right? So here's fungal pneumonia. This is an extremely severe case of fungal pneumonia. This patient, I can't imagine, was living without some sort of assistance respirator of some sort. But here's a CT scan of a healthy lungs, and then here is a fungal infection in the lungs. That's a mold growing in the lungs. So they can damage through direct invasion. Here is like histological slides of the integument of the skin. So here's the epidermis, here's the dermis, and here are, remember spaghetti and meatballs? So here are if this is a dimorphic fungi, maybe they're not, it wouldn't really be a dimorphic fungi. This would be yeast cells and pseudohyphae, or if you zoomed in more, you'd be able to tell more that these are what are called the fruiting bodies. So I'm not 100% sure unless I'd look at it under higher magnification. But you can see the invasion of the tissue is the point. So it's invading the skin. Okay, so another methodology of CPE for fungal organisms are toxic enzymes. So even though bacteria make these too, we didn't talk about these a ton with bacteria, but fungi, we do talk about them. So they make these enzymes that will degrade tissue. So remember we talked about the pizza and how the bacteria would spit stuff out, digest the pizza, and ingest the pieces? Well, fungi do the same thing. They spit stuff out. They spit out these toxic enzymes, digest what's around them, and then they'll be able to eat the remaining pieces. So those are just toxic enzymes. So these are two examples that I'll give you are proteases and lipases. So remember, ACE means enzyme. Protease means it's an enzyme that breaks up proteins. Lipase is an enzyme that breaks up lipids or fats. So it can come up with all kinds of enzymes that are toxic to their human patient. Okay, and then we have another whole category that are called mycotoxins. So these have their own special branch because even though they are toxic, they're not enzymes. They are toxins that are like extremely toxic. So these aren't just like local infection, something, so it's actually a toxin that can cause your patient to go into like toxic shock. It's like they're extremely toxic. And there are all kinds of different kinds. There are many kinds, hundreds of different kinds. But there's only a few fungi that will secrete mycotoxins. They're more severe than they're more severe than secreted enzymes. They're severely toxic. So one that we are going to remember that I will have you memorize are aflatoxins. Because I have a good example of what that is, and then it will just help you remember what mycotoxins are. So here, this is aflatoxin. So aflatoxin is a toxin that is spit out by some species of Aspergillus. And so there's an example of so there was a researcher, he worked for a prominent university in England. It was like King's College or something. I don't remember where he worked, but he was trying to solve the food crisis in parts of Africa. So he was like, oh, when he was in his 30s, in his early career, now I think he's 80s or something, he's old now. But he was young when he first started out trying to solve the food crisis in Africa. So he first started out saying, okay, well, let's import a bunch of cattle and goats and things like that. And so then they can survive indefinitely on animals. Well, this is always the problem with trying to help when you don't know anything about the place that you're bringing those animals into, is because animals eat 3 times, 5 times, 7 times as much as humans do, and they need way more water than humans do. So is it better to have 1 cow and feed 1 cow, or to feed 7 families from the food that you have to give that cow? So that didn't work. Even though still from his intervention that there are many African farmers, because of his intervention, that still are trying to get the animals to live off the land, even though the land is arid in a lot of parts of Africa. So his intervention, he tried it, it didn't work. So then he said, well, the elephants are trampling down all the grasses, and the grasses are needed to keep the water table up higher. So he's like, let's kill all the elephants. So he went in and they slaughtered and rounded up thousands and thousands and thousands of elephants in different areas of Africa, so that the cows could eat the grass and it would pull up the water table in the areas, so that the people would have more access and the animals would have more access to the water. Well, after he did that, they figured out that actually the elephants were hugely beneficial for the grasses, because they would stomp all the grasses and prevent erosion and prevent the sun from burning straight to the soil that got too hot, that would kill the grasses from the roots. So all the grasses died anyway. He killed all the elephants, and this animal still wouldn't survive. The people were still starving. Not a solution. So then he said, okay, well, let's try and grow another crop. I mean, you got to give the guy props for trying, but geez, like, come on, do a little test run in one little area, like do something. I don't know, like, okay, so then he planted peanuts, which are indigenous to some parts of Africa, but not other parts of Africa. But he's like, let's grow peanut crops like crazy, because peanut butter is a high caloric food. Let's just try peanuts. Well, what happened was they grew aspergillus all over the peanut crops, and the people that ate these peanuts and peanut butter ended up with mycotoxin poisoning, with aflatoxin poisoning, which these toxins are so poisonous that they can cause cancer. So they're cancer causing, and there was hundreds of children and families that ended up with liver cancer from aflatoxin poison from aspergillus that grew on the peanuts. So yeah, that was the whole spiel behind aflatoxin. So mycotoxins are still, we worry about those a lot all over the world. And so mycotoxins are, we worry about that a lot in pet food, because pet food is the regulations around how we make pet food and how we grow pet food. We have lower regulations than animal food. So it's under less rigorous control. So they worry about that a lot in dog and cat food as well. Yeah.
Is all aspergillus black mold, or is there just certain ones that are?
No, is all black mold aspergillus?
No, is all aspergillus black mold?
No, that's a great question.
Okay.
Yeah, there's different types of aspergillus that can cause different things. Yeah. Good question. Okay. Okay, so here's another mycotoxin, but we don't need to remember this one. I just wanted to let you know about, so these are the traditional mushrooms that grow in the yard, right? These are what are called death cap mushrooms. So death cap mushrooms have a toxin, a mycotoxin, that's so severe that 9 out of 10 fungal related deaths are attributable to these mushrooms. But it's highly virulent, the toxin. It's extremely deadly, but not very many people will die from it because they can get treatment. And so just be careful of statistics like that as well. I like to kind of throw that out there. When you read a statistic, think about what it really means. Think about like the alternative. So if you said 9 out of 10 fungal related deaths are attributable to the death cap mushroom, then you'd think like people all over the world are dying from these mushrooms. Well, out of fungal related deaths, how many people have fungal related deaths? How many people have? Not very many. So, yeah.
Of these 7, is this like from direct consumption, or are there like environmental ways that it could affect things that then?
That's a good question. I don't think it can be environmentally leached, but that is a good question because it seems like it would be an environmental hazard, but I've never heard of that. That is a good question. Okay, so yes.
Are there certain parts of the world where you have to worry more about funguses or molds, or is it? I mean, I assume there are. But...
Wonderful question. Yes. So, parts of the world, what do you guys think that are more susceptible to fungal infections?
Humid.
Where?
Humid areas.
Humid areas.
Oh,
Yes. Moisture. So they do need. They do prefer an aqueous environment. They do, not that they need it, but they, when you hike through the forest and it's like wet old logs that they're decomposing. So they're saprobic, they're breaking down those trees and soil and all that kind of stuff. And it's usually in the shade, so they don't like direct sunlight, and they like a little bit more moisture, and they like heat, they like warmer temperatures. They don't like cold very much, but some do. So it depends on the species, but generally, just broadly saying, it is more like in the southern states have more problems with fungal infections, more so maybe Southern California. Even some places like along the equator are too hot, or along the equator are too moist, so they need this like special medium environment. Yeah, great question. Okay, so here is what can happen with fungal infection. So I just put FYI on here, but we're going to talk about this a little bit more, and we're going to talk about how this affects us when we talk about our Aspergillus nocarb. But it says 70% of homes have some kind of mold in them. I know my house does, so underneath we have like a crawl space, and underneath that crawl space is full of like white mold, which grosses me out, but they said it's normal, so I don't know, it freaked me out for sure. I was like, get rid of it, but it's wet under there almost year round, even when it's really hot, so what do? So yes, it's always, you have to be really careful if you see black mold in your house at all, and chronic exposure can cause huge problems, and we'll talk about that when we talk about our DDX section about chronic exposure, because most of us that are immunocompetent are fine, and if we don't have chronic exposure, we're fine. So I'll talk about why. Okay, so let's talk about repro. So I'll just put a couple little stacks and have you guys pass these around. So I made these little coloring pages, so I'm going to give you an extra credit point on your quiz if you have them colored when you get back for your quiz day on Monday. So don't forget. So somebody that goes, I forget, can I have extra quiz point? No, it's just extra credit. So just do it or don't do it. But it doesn't have to be beautiful either. So just the point of coloring it is so that you see the different structures and that you color label and color code the different structures. So if you color code the different structures, it'll help you to understand them a little bit better. Do you guys have oh yeah, there we go. Cool. Okay, so just like Dr. Duffy was saying, was why do we care about fungal repro? We care about fungal repro because we think about it when we are, you know, more so like the CDC would think about it, or the NIH or the World Health Organization would be concerned about temperature patterns and things like that. But you would know if you're a physician or a nurse practitioner or a registered nurse or a medical assistant or whoever working in Georgia, you'd be like, oh, it's fungal infection time. Oh, 3 of my patients came in today with a chronic cough. What's going on? And you'd know it was seasonal if you lived in that area. Yeah.
What is the green and the red?
So
Represent?
these are just different species that will grow in different areas based on different temperatures, but I just kind of threw that on there because it's just fungal infections that grow in different areas at different times. Okay, so let's talk about yeast. So just so you know, when we talk about mitosis and meiosis, I think it might be easier for you to think about it in the case of human beings. So it helps us to understand our own bodies a little bit better, that we understand that every cell in our body is diploid. So every cell in our body has one chromosome from their mother and one from their father. In all of our cells, eyes, ears, nose, liver, kidneys, all of our cells have 46 chromosomes, 23 from our mother, 23 from our father. And then our gametes, our eggs and sperm, have 23. So they are haploid. They have half the number. So they have 23 in every egg has 23 in it, or every sperm has 23 in it. And that's because when you meet the 23 with the 23, it makes 146 organism. But when you talk about fungi, they get really weird. They get you have to know, just shit gets weird. You just kind of run with it. Just know the process of mitosis, know the process of meiosis, and then just know fungi are just weird and can kind of do anything. So asexual, they can undergo asexual reproduction. So one yeast can go through, we would still call it binary fission. They'd go through binary fission or asexual reproduction and create 2. But the funky thing about yeast, I wish I had more space. The funky thing about yeast is you can have one yeast here and one yeast here. These are both 2 different yeasts. This one can have, I don't want to use N and 2N, but let me put those to the side just so you know. So this yeast can be diploid, this yeast can be haploid, and then it can go through binary fission, and this yeast can have 2, and this yeast can have 1. They can each, so a haploid can go through binary fission and make another haploid. A diploid can go through binary fission and make another diploid. So it's like if an egg from a female in the ovary were to just sprout a whole human. It needs that other set of chromosomes to be able to sprout a whole human. So remember, fungi are weird. They can be haploid, they can be diploid, they can go through binary fission, do their own thing, and be their own yeast living in the world. Okay, so this process is called budding, where the little bud comes off, but also, just so you know, it's called asexual reproduction, which is also generally called binary fission. Okay, but also they can sexually reproduce. So a haploid and a haploid can mate together and go through meiosis and can make a diploid organism. So the haploid and haploid can make a diploid. Yeah.
Do the number of chromosomes of the 2 cells that are combining really matter, or can it be like one of them has 2 chromosomes, one of them has 1, and they combine and make a 3 chromosome
Generally
fungus?
they don't. Generally they don't create a different number other than the half and the whole. but some organisms can do that, like frogs. A lot of plants can do that. Some plants, there's a term for that. That's called like, I forget what the term is, but there's a term for, it doesn't really matter how many chromosomes they have. They can have 20 or they can have 5, as long as they have the right number of chromosomes that make the right number of proteins to make the organism. The extra chromosomes don't do anything. But in the case of yeast, generally there's a haploid and a haploid or a diploid. And probably it's because the way that they pair up, and one, they have to have the partner to be able to make the correct amount of proteins to be able to form a new organism, is my guess. Yeah?
Do you know what, like, evolutionarily causes an organism to have a certain number of chromosomes? Like, why do we have 46 and a frog might have 5?
Yeah, no, I don't know that. That is a really good question. I'm not sure why evolutionarily, what the point behind the number was. If it's, I wouldn't imagine it's accidental, but some of the genome, the genetic material, a huge portion of our genetic material, we don't even know what it does yet. So, like for a banana, for example, they have more chromosomes than we do. Then it's like, well, we don't know what all that extra chromosomal material does.
Right, I know on some of our chromosomes they hold like way more important information than others
Yes,
do. So,
yes.
like,
But
why is it
ours,
on
we
that
know
one?
you can't jack up. We know if we're missing a piece of any of the two between a human, if they have any sort of large scale, like any sort of base pair missing more than that is associated with one amino acid change, then we have big problems generally. Yeah. So you would need both pairs in humans, but not in yeast. Yeah.
Is there a correlation to like the success of an organism?
I would say no, because like flowering plants are hugely successful and they have huge chromosomes. But chromosomes, if you could say generally, you could say like humans are, you wouldn't say humans are more successful than flowering plants, because there are more flowering plants than there are humans on the planet. But I mean, I guess it would depend on what you mean by. Yeah, I would say there's not really a correlation. I don't really know. Yeah. So, but the one thing I did want to bring up, that's a good point, is that what is the point of making a diploid organism? Why, if you can make a haploid organism, it seems much more complicated that to make a diploid organism, to find a mating pair and to share genetic material. Well, it's the same reason I brought up for the cheetahs. So remember the cheetahs, that they all have the same genome, essentially, where they're all so closely interrelated that you can give, you can do a blood donation from one to another. You don't have to type across them or anything. Like humans, you would kill a patient if the patients weren't the same. So, but cheetahs you can, but the same problem with yeast. If you have a haploid yeast living on, and then it gets some bacterial infection, or it's got a temperature variation that it doesn't like, or something goes wrong, but just the genetic, there is always an evolutionary advantage to genetic sharing. And that genetic sharing causes variations in the population, and those variations in the population can be beneficial because that will help prevent disease, and that generally that's why organisms that sexually reproduce have been highly successful, is because of genetic variation causes protection against all kind of environmental adversities. Okay, so here's like a little picture, if this helps you, that there could be a haploid that can reproduce, there can be a haploid that can reproduce, or those haploid and diploid can share chromosomes and form a diploid organism, and that diploid can go and reproduce and produce haploid gametes, or it can go through binary fission itself. Yeah.
So when the two haploids form together, are they literally squeezing into one cell, or are they just exchanging DNA?
With yeast, let me think. So for molds, molds, what they can do is they can swap DNA. But yeast do form one structure. Like if it's a haploid and haploid, they form one structure. I think they can do that. I'm
Okay.
trying to think back on my.
I'm just trying to picture it in my brain. Like,
Yeah.
so they like two balls to one ball, or is it like they like exchange they go through mitosis and then exchange it?
Yeah, I'm trying to think back in my own studies if yeast are able to do this and the chromosome comes out.
Yeah.
And I don't think they can. I think that they have to share cytoplasmic membrane, and then they form a 2N, and then that 2N, I think one degrades, and then this 2N can form another 2N or can form a 1N, 1N.
Okay.
Yeah.
Okay. Thank you.
I'm thinking if I can remember that quickly. Okay. Okay, so reproduction in molds. Okay, so molds are really weird because they have spores. So just so you know, everybody pay attention for a second. They're not endospores. These are not endospores. Endospores are a structure formed by mostly gram-positives, and they're bacteria. Totally different. Spores, if you think of what spores are, they're more like pollen of plants. What they are is they're a little sperm or egg floating out in the ether. And they're not endospores that are hardy, peptidoglycan-filled, environmentally resistant bacteria. These are little pieces of sperm and egg that float around in the environment at any given time. So that's why the strawberries maybe did come with fungal spores on them, but say you bleached them, like you wouldn't bleach your strawberries, but say you bleached your strawberries as well as you could, washed them as well as you could, and left them out on the counter, in 3 days they'd have fungi growing on them again. Because they're everywhere. Spores are everywhere. They're in our lungs, they're on our skin, they're in our hair, they're everywhere and on every surface all the time. And we might not see them, but if they're making every plant out there grow, there has to be fungal spores everywhere. They're just all over the place. So even though we can't see them, they are growing. So for example, the yeast that I grow, the way that I make sourdough bread, so I have wheat and water. And this wheat flour and water, I can mix it together and cook it. It'll come up with a really, really flat, hard pancake, which I've made before. So that's like a brick and a paperweight. But if you let the whole wheat flour and water sit for 2, 3, 4, 5, 6, 7, 8 days, then it starts getting huge and bubbly and starts smelling really funky. And that's because it has yeast already in it, and yeast already on every single one of those little wheat flour pieces has yeast in, on, and around it. There's yeast everywhere. So what's happening is I let the yeast start eating that flour, and they need some water, they need a little aqueous environment, and so what they're gonna do is they're gonna start living and growing, and that yeast lives and grows, and the yeast start forming CO2, and that's why it starts getting bubbly. And the yeast start fermenting, going through anaerobic respiration and producing lactic acid, and that's why it smells funky. That's why sourdough bread is sour. And so that's what you're doing when you're pouring in the yeast packet. Then you have Saccharomyces cerevisiae in that little tiny packet. You cut the packet open, you dump it in there, you mix it with some flour and some water, and in that case, if you put it in a warm environment like yeast like, it'll double in size in 4 hours. You'll have a flour that's this big to flour that's this big. And it's huge, although it was just, you put in a packet of yeast, it's because that dehydrated yeast, you're putting in such a huge concentrated amount of yeast versus letting the yeast grow over time. That's the difference between sourdough and regular bread. Regular bread, you're putting the yeast, so much yeast in there, and they're not making very much lactic acid. They're making some lactic acid, but they're just growing and thriving. But if you let them grow over time, the lactic acid builds and builds and builds in that environment. And so that's what makes sourdough really sour. Okay, so when we talk about molds, molds can do all kinds of funky things. So just remember, they can do all kinds of weird stuff. So let's talk about asexual reproduction. So asexually, they can have their hyphae. I'll show you a picture of what this looks like better. So remember, we have vegetative hyphae and aerial hyphae. We have hyphae is one, or some together, and then mycelium is a bunch. So here's mycelium of a mold. Remember, a mold is still a fungi, it's just in a different shape than the yeast that come in only like little round shapes. So here's our mold. Here on the top we can have sporangiospores in here, or we can have conidiospores like this. And they're just out and on top like that. So I need my tall person again. So what molds can do is they can have this little area of hyphae can split off and create a whole nother organism asexually. This little hyphae piece can split off and form its whole own organism. Or a spore can come over and land over here, and if the environment was right, it would form its whole other organism.
But the organism would be a different number of chromosomes than the original?
It can be. The same with yeast, it can be a haploid or it can be a diploid.
Okay.
Same. With
Does that
yeast.
depend on whatever came off of
Yeah, whatever came off and what the
Whatever
environment
cell
was,
started it.
whatever cell started it, yep, perfect. Whatever cell started it, and then what the environmental conditions were like in that, were there enough nutrients? You know, those spores are so microscopic that they can float in the air. And the reason that they want to float in the air is so that they can drift off and further away from its original source, so maybe they'll have more access to nutrients. So I think about that a lot with, like, you know, fruit. The evolutionary advantage of a plant making its own fruit was so that an animal would eat it because it tastes delicious and run off somewhere else and poop it out. Well, now the poop, it already comes with its own manure, it already comes with its own food source, it already comes with its own, it moved further away from its source, so now maybe it'll be successful over there and grow another plant. So that's kind of how spores work, is they're so microscopic and they can float in the air so that they can travel at different distances and be more successful everywhere. Okay, I have lots of rants today. Okay, so hyphae can fragment off. This is called fragmentation and become its own organism. So hyphae can fragment. And then when we talk about sexual reproduction, so it can be spores. So spores can go and do their own thing. Hyphae can split off and go do its own thing. And then they can also sexually reproduce. So they can, one branch of a hyphae can actually fertilize another branch of a hyphae. So this little DNA segment can come and fertilize another area of the hyphae. Yeah.
Of the same plant?
Even within the same plant, if
Well,
it's not
then it's
diploid. Yeah.
Then
So-
it's not going to be different if it's the same plant, is it?
So it will become diploid. So some cells that are diploid can make they can make gametes within the own plant. So not all cells within that fungal organism are haploid or diploid. So some parts can be diploid, and then they can form haploid cells. And if that haploid cell shares its DNA with another haploid cell after it's undergone replication, then that DNA can spread. So it can basically like replicate its genome and share it, replicate its genome and share it. So it can fertilize if there is another haploid area of that plant. So they can share genome within themselves and they can share genome within other plants. So these, they have like male and female parts. The male, the reason they call it male, they don't have male and female organs, but they call it male and female because they call the male the giver of the DNA and the female the receiver of DNA. And I know that this seems like we're getting into semantics of why do we care, but actually mycologists think about this a lot because there can be an organism, like some sort of fungal infection within one patient can have a different genome within one patient. So that one patient can have like 3 different strains of the same fungi within that one patient because they can share genetics within the patient themselves. So they do worry about it a lot. So the section of the hyphae that gives the DNA is called the antheridium. The section of the hyphae that receives the DNA is called the oogonium, or oogonium, antheridium and oogonium. Sometimes it's called an oospore. That's the sexually recombined spore. Now it's called an oospore, which is similar to how we name it in humans. So antheridium is the male, oogonium is the female, oospore is the resulting diploid piece of the hyphae or spore that's going to go and make its own plant. Are you with me? Kind of does all kinds of stuff. Yeah,
So are they on the same, like, fungi?
it can be. It can be on the same fungi. If it's a haploid and haploid piece, it can share its haploid genome. If it's on a different one, it can share between 2 different fungi as well.
So it's gonna reproduce with itself?
With itself,
Okay.
yes. Yeah. You can almost, like fungi, the way it works, if you think of it maybe in this way, is that fungi almost, even though all of their cells work together to make one multicellular mold, each cell is almost like its own individual organism. They just work together, they just fit together and clump together. But really, each individual cell has the ability to make its own organism. So fungi are just weird, just roll with it.
And the spores like land next to each other.
Spores, yes. Yeah, hyphae can do that and spores can do that. Yes, yes. So what the spores can do also is, this is kind of a cool, scary / thing that spores can do, is not really spores, let's talk about hyphae specifically. So when the 2 fuse together, I guess you could call those spores, but just don't focus on the semantics. Because they're individual pieces that are coming off of the hyphae, and these individual pieces can come together to form this new weird thing. And what this new weird thing is called a zygospore. So this zygospore is a sexually recombined spore, and why we care about these is because it's almost like an endospore. They're crazy environmentally stable. They can withstand freezing temperatures, drying, they can withstand, this is how fungi can grow even though like the soil's been depleted for 100 years and it hasn't had any moisture, and then it rains once and all these fungi come up. It's because they have these things called zygospores that are environmentally resilient. So in a patient, if a patient has a mold infection and those 2 hyphae formed these little pieces together that are called a zygospore, now your patient has something that the pharmaceutical will never get into that. The pharmaceutical won't. You'd have to kill everything else, wait for that zygospore to form another fungal infection again before you could try and kill it with an antifungal. Yeah.
Are these related in any way to endospores?
No, they're not related in any way. The only coincidence that they have with endospores is that they're called the same name. And then the only thing that they have a coincidence with endospores is that they're environmentally resilient. Yeah, yes. But other than that, they don't have any similar structures, anything like that.
So like endospores will kind of go into that kind of dormant phase where they just don't really do anything, and
Yes, zygospores too. Yes, yeah. So zygospores can withstand a lot of environmental, so other than, the difference between zygospores and endospores is endospores, it's just a totally separate organism that can do totally separate things. Whereas zygospore, it's more like if you think of like a seed. Seeds from a plant can withstand all kinds of different temperature variations. You could store seeds for hundreds of years. You buy packets of seeds in the store, and you could keep those on the shelf for however long they'd be good for, and then you put them in the soil and they grow. So it's almost more like a seed. Yeah.
should we just kind of think about it that endospores belong to bacteria and zygospores for fungi?
Yes. Yeah. I just don't want you to confuse the two because they're they're from two completely separate organisms.
I'm having a hard time figuring out, the fertilization and the fusion seems so similar.
Yes. So fertilization is exactly what you think of it in humans. So fertilization is a male and a female DNA shared. And then they can also undergo fragmentation. And that fragmentation is like if I cut off my hand and put it in the soil and it would grow another me. So it's not any DNA sharing, it's just a piece that was cut off and will grow its own organism.
Oh, okay, so in fertilization, the new cell splits away from the original plant?
Yes, and that cell can become its own plant, and its own diploid plant.
So the oospore separates from the original plant, but doesn't the
Yes.
zygospore also separate from the original?
Yes, so a zygospore is an oospore basically, but
It's
it's
just-
one that formed this really weird structure.
Okay, so the
Yeah.
difference is that it makes a strong structure.
Yeah, it just makes a strong structure.
Okay.
Yes, yes. Everybody with me still? Everyone okay? Okay. So here is a microscopic view of a zygospore. I will show you that in the lab as well when we get to this section. But it forms this environmentally resilient. So if your patient has zygospores in their lungs, they're sad. So they're a sad patient. Give them lots of hugs, and then you're just cry. Because what gonna give them antifungals, and there is no way an antifungal will get through that material of the zygospore. They won't.
So do we know whether that would like cure?
So what happens when they form a zygospore, usually what they'll try and do is give the patient antifungals and kill off as many as they can, and then they have to give them series of antifungals so that when that zygospore will sprout, because you killed off everything else, so now there's nutrients around, now there's space, now that zygospore can form another one, then you give them antifungals, then it will grow, then you give them antifungals until hopefully you get rid of the whole
So they're gonna have
population.
a fungal infection for a very long time.
Yes, and most people that have fungal infections do have fungal infections for a very long time. Antifungals are very hard they don't work very well. Sometimes it'll take months. I came home the first time from Africa with tinea capitis, it's a fungal infection in my hair from carrying kids on my shoulders. And so I came home with it and I was like, oh my gosh. First of all, I hate hair stuff. My daughter had lice and I had nightmares for months. I went and cleaned everything up and lived in a hotel. I was like, I can't do it. I'm done. So I hate hair stuff. So I had tinea capitis when I came home and I was like, oh my gosh, I have a fungal infection. And then my niece sent me like 100 pictures of me carrying kids on my shoulders. I was all, that's my okay, don't do that anymore. So so anyway, what I had to do is use antifungal shampoo. Now we're all like itchy, huh? So I had to use antifungal shampoo, and it took probably 4 months of antifungal shampoo. Just antifungals are hard to get rid of, and so we'll go through the diagnostics of why.
But like something like monostat that's used like for yeast infections, it seems to work like so well.
Yeah, it does. Actually, yeah, for yeast infections, they do actually, the antifungals do work really well. And what can happen is once you get rid of that fungal infection, usually the healthy bacteria can take over. So usually the healthy bacteria will act as an antagonist and help get rid of the fungal infection. Because really, even though candida and all kinds of different yeasts live in the vagina, they're not the primary organism. And so when they start taking over as the primary organism, as soon as you kill them off, the other organisms do better.
It doesn't usually, never mind.
Mm-hmm.
Never mind.
Okay. Yeah, go ahead.
So for the oospore,
Yeah.
it's created via male and female
Fertilization. Yep. Perfect.
And then so for the fusion, it's also, you say it's also male and female, but it's just something else happens that makes it a zygospore?
So I can't remember, when did we talk about fusion? I can't remember where
It's
I brought
option
that up. Option 4.
Is it?
Oh, so fusion is specifically fusion is to make a sexually recombined spore. The zygospore is the actual, fusion is different. If you think about it, about the term, the fusion is when they fuse together versus when the fertilization occurs from one to the other.
Okay,
So look at the fertilization one where it's like, and then the fusion one where they come together to form one individual spore structure.
Is the spore structure that H-looking thing, or is it just
This
the
thing
pigment?
right here,
Okay.
that, and then it will form the zygospore around it.
So D is the zygospore. Looks like.
D.
Well, it's labeled D
here.
right there. Yeah.
Oh, yes, here D is the zygospore, yes.
Okay.
Yeah, so hopefully it will help you as you're doing your little coloring page, because it does a good job. I just pulled this out of a micro textbook, but it does a good job, like kind of labeling each little part. And so just color those, and don't worry about the ones we didn't talk about any of the terms we didn't talk about. But it will help you to like identify what those little structures are.
So the hyphae are not part of that,
The hyphae really like they branched off and fused together to make the zygospore.
Okay, so the whole blue circle is what we would consider the zygospore?
No, just this little piece. Those pieces will fall off, and you'll just end up with this little thing. This is a zygospore, but really this can go and do its own thing.
Okay, the hyphae
Yeah.
and all of them.
Yeah.
Okay.
Yeah.
What would happen what happens when your fungal infections go untreated? Like, for example, if you didn't treat your hair with the fungal shampoo, what's gonna happen?
Oh, usually my immune system would fight it off if I was immunocompetent. It would go away eventually. So a lot of kids do you'll see tinea capitis in kids. If you look now, you'll notice what it is. So you'll see it. It looks, it's like sloughing skin. Babies get cradle cap. Yeah, and cradle cap, it looks like that, but tinea capitis can sometimes look orange too. So, once I had it, then kids all over, like my daughter's swim team, I'd be all, you can see it, it's pretty common. But yeah, it's just like sloughing
What
skin.
about like the one in the throat with the zygospore that forms? Or do you need antifungals to treat these, or will they all eventually go away?
That is a really good question. So if there's if it's topical, most people would use a topical antifungal to get rid of it. But you don't always have to, your body will get rid of it as well. Like ringworm, for example, is common in kids, and many parents will go get an antifungal cream and clean it with the antifungal cream, but some parents don't and just leave it, and their body will
And the
eventually get rid
example
of
with
it.
the lungs, which was obviously a much more serious condition.
Yeah, if there's a fungal infection in the lungs, it's IV
Because it's
antifungals.
inhibiting other things your body needs to do.
Yes. And yeah, so skin, we don't worry about as much, and it can be topical. And like vaginal, usually it will go away on its own as well, but usually patient is uncomfortable and will have to go in and get treatment. But anything, in an immunocompetent patient, generally your patient will be fine.
So we
But in a-
worry about bacteria much more than fungal.
You would, unless your patient is immunocompromised. If your patient is immunocompromised, you worry about fungi a lot. And so I'll tell you why. Like, why would you worry about fungi versus bacteria? I'll see if you guys can come up with that one. Yeah.
Okay, so I have a clinical question, and I don't know that you know this, but I'm just curious. If somebody has an infection under their toes, it's usually fungal, right?
Yes,
Under their
yes.
toenails?
Uh-huh, anicomycosis. You guys will
And
learn
if
about
it's
that.
under the toenail, does it go away on its own? Or if it doesn't, does that mean they're immunocompromised?
So skin infections, skin fungal infections can happen no matter what. So like kids, yes, they are immunocompromised, they are more subject to ringworm. But if you touch a kid with ringworm, generally an adult will get it. But
They
like,
will?
They will get it. An adult will get it. But not, probably, this is 0% accuracy on my part, it's just anecdotal, but I would say 80% of the time an adult won't get it, but 20% of the time they will.
Okay.
But adults, but if, for example, like my husband has athlete's foot, he showers at the same shower as all the other firefighters. It drives me nuts. It grosses me out till no end. So I gave him like shower slippers. These are your shower shoes. Like, do not wear those. Okay, now you have to wear your shower shoes at home because you won't wear them at work. So it grosses me out till no end. So I clean my shower religiously, but at the same time, I would have gotten it. You know, like we share a shower. We walk barefoot in the same house. But I don't get it for whatever reason. But he will get it. So in the diagnostics of our fungal infections, which we'll talk about, are sometimes it's also with fungi, it's chronic exposure. So if you're exposed to it over and over and over. But to answer your anicomycosis question, so anicomycosis is when the nails turn yellow. So some people have it in their fingernails, some people have it in their nails, in their nail beds. And just so you guys know, everybody in this room will have it as you get older. Everyone gets it as people age, generally. I would say the majority of people in the world, as they get older, will get a fungal infection in their nails as they get older. And that's because the tight junctions between our skin just starts getting more loose and more loose. There's moisture in our socks, there's moisture in our feet. Fungi like that, and they like a warm and moist environment. So a lot of people will get a fungal infection in their nails as they age 60, 70, 80 years old. So yes, it is because of age, but is it immunocompromised? I mean, because of the lack of cell tightness anymore, like is that immunocompromised? Kind of, maybe not, but most people will end up getting it just because the cells break down over time and we lose that tension in our skin.
And there's no way to get rid of it at that point?
Well, so some people that will get it when they're younger, like getting a pedicure, then they can get rid of it, use antifungals religiously, keep feet dry, use powders to keep feet really dry and take care of it. It'll take a long time to go away, but it will go away. Older people, how long can you keep your feet dry? How long can you use antifungals for? So a lot of times in older patients, people are just like, man, live with it, you're fine. Yeah. Okay, so I'm not gonna talk for too much longer. I'm just gonna talk for a few more minutes and we'll get into antifungals and DDX in a little bit, but let me just finish off on these last couple slides. So let's talk about transmission. So I put here mycotoxin transmission, but it can be transmission broadly also. So just kind of think about it in fungi in general. But how we get mycotoxin poisoning or how we get a fungal infection. So we talk about those two things kind of separately because you can get a mycotoxin poisoning without getting the fungal infection itself just by eating the mycotoxin itself. So that is something to worry about. So almost like Dr. DeLeonards was saying, like, can you get it from leaching in the soil? I haven't really heard of that, but I know that you can get mycotoxin poisoning when there's a giant concentration of mycotoxins in the food, for like dog food or whatever. So, humans can get mycotoxin poisoning or a fungal infection directly, or we can get it indirectly. So this was a good review, our biological vectors versus our mechanical vectors. So our biological vectors, we can get it from meat, dairy, eggs, from animals that are infected with fungal infection, and then their meat or whatever is rendered, and that is a high concentration of those fungal toxins or of the fungi itself. But also we can get them directly from the crops if we, that's where they got it. They got it from the crops, or we can get it from the crops. So if we eat the plants also that are infected, that's why we have we have a very rigorous process of farming and how food is treated and how we're able to sell food and process food because of fungal infections, or from the fungi itself. So look through this and see like kind of how those are passed around, and just give yourself a little review of direct versus indirect, and those biological versus mechanical vectors.
I can't find the slide. Where are you at?
Oh, I'm sorry, I'm on the new slides. I'll just go over a
for
couple
the
more and
next
then I'll post the new ones. Yeah,
Oh,
these are number 17? 17.
So you just haven't posted them yet?
Yeah, I haven't posted them yet.
Okay.
Okay. I just wanted you to see what fungal growth looks like. We already showed you that. Or it can be fungal growth with mycotoxin, where the toxin will actually be degrading the tissue. And then when we get to diagnostics, let me just introduce diagnostics, and I won't ask you about this on Tuesday yet, but I will for the next following quiz after that. But let me just discuss diagnostics. So bacteria are so different than fungi. So we worry about fungal infections primarily for immunocompromised patients. That's when we really worry about it. So say your patient has HIV, you have two patients with HIV. One patient, both patients you say stay home, watch out, you're immunocompromised, be careful. So why do you worry more about fungal infections in HIV patients than you do bacterial infections? They're already immunocompromised. But then what is going on with transmission? Like how as an HIV patient, like what can you do to prevent infection?
You can't really.
Great answer. You can't.
Yeah.
You as an immunocompromised patient, you cannot prevent fungal infection. You can't clean enough. You can't disinfect enough. You'd have to live in a bubble, right? Because there's fungal spores everywhere.
But isn't it the same with bacteria or no?
Well, bacteria, yeah, they can be on the dust and things like that. But basically, if you're at home and your house is pretty clean, then you won't get a bacterial infection. Especially if there's no other people in there, then they'll die. But you can't keep fungal spores from out of your home and out of your windows.
Okay.
So fungal spores are just everywhere, and so patients that have HIV, for example, patients that are taking immunosuppressants who have had liver transplants, heart transplants, anybody who's on immunosuppressants, patients who are on chemotherapy, so chemotherapy will greatly reduce the number of white blood cells, so your patients who are immunocompromised who are going through chemotherapy. So what will happen is generally HIV patients or immunocompromised patients will get numerous fungal infections. They'll get nail fungal infections. They'll get thrush. They'll get skin fungal infections. They'll get vaginal fungal infections. They'll get all kinds of fungal infections because there's nothing really they can do about it because we can't prevent it. It's everywhere. Yeah.
So what you're saying is if there ever was a fungal zombie infection, there would be nothing we could do to prevent it from killing us all?
That is a fantastic wrap back around. I love that so much. Yes. Because really that was what I wanted to come right back around to that topic. So perfect. Is because really what could you do? Like how do you get rid of air? Like how do you get rid of fungal spores? It's like getting rid of air, so we would have to come up with some crazy antifungal, and antifungals don't work that well, and what would you do? So, yes, that is, now that I've thoroughly terrified you, yes, there is not really much we could do about it. So, fungal organisms are like the perfect, the perfect example of how a patient can get an infection from being immunocompromised, because they are so commensal. They live everywhere and around us, and because we're immunocompetent, our bodies are fine with fungal spores. Our bodies deal with them every day, all day long. Our immune systems are fine. Our immune systems will recognize them. Remember I told you the story of the girl with the peanut butter, that she had the anaphylaxis within 30 seconds? So that our immune systems within 30 seconds are used to dealing with stuff constantly, and our bodies will fight that off. But as soon as you drop our white blood cell population, then everything is a pathogen. So, but our immune systems, even though we are immunocompetent, the problem with things like black mold is that people live around those spores so often, and their immune system is constantly overloaded trying to fight off just being inundated by those fungal spores all the time. Then they can start getting headaches, then they can start getting nausea, then they can start getting fatigue, and that you'll see these signs that your patient maybe has been exposed to mold. So, and then they can look for black mold in their house. So for example, I have one girlfriend, her name is Jess, and she has 3 kids, and her and her 3 kids lived in an apartment, and they started getting headaches first, and then all of a sudden they were, all 4 of them, like deathly afraid of gluten. And they were like, why did all of a sudden all 4 of us, now we have celiac where we didn't before? Like, what is going on? So they went to specialists, and then they found out that there was black mold in their house. So it was inundating their immune system so much so that it was making them immunocompromised. So now even wheat were having a reaction to their body. They were having headaches, they were getting sick regularly. And so long-term exposure is a huge problem for people that work in fields, or that work in demolishing houses, or work with animals. Animals have a huge problem with fungal infections, and working with animal feed, working with birds, like bird feces grows fungi on it really easily, or any animal feces basically. So anybody that has long-term exposure to high risk areas will also be at risk for a fungal infection. Yeah.
So once they got away from the black mold, was their tolerance for food better?
Yeah, no, it's forever. So now they're all celiac. They still have all kinds of immune problems for now still years that they've all still have immune problems. So the mother was better, like her immune system bounced back a little bit better, where she started feeling better pretty soon after they moved out. But all 3 kids, it took them years before they started really feeling better, and still they all have immune issues. And yeah, so it's a problem. We worry about it a lot as clinicians. It's hard to diagnose, because you're like, oh, headaches, malaise. Well, you're under high stress environment. Go home for a month. I still have it. Oh, maybe it's your food. Go home for a month. Oh, maybe, and so it's hard to recognize and diagnose.
So are there any physical traces of the black mold in their bodies, or how do
How
you have
do
to
they
check their
diagnose
house?
it? Yeah,
Or how do you check do you just have to check their environment?
Yeah, you just check the environment.
Okay.
Yeah, so they have experts that come out and check your house for black mold. So they'll check under the toilets, they'll check under the house, they'll check in the attic spaces between the walls and things like that, if there's dripping and black mold showing up. Yeah.
So with the case of Jess and her children,
Yeah.
in the apartment, is there any specific portal of entry, or is it just where is the black mold actually affecting you? Are you
They're breathing it. They're inhaling spores.
And
Yeah.
so
Good question.
like a mask probably doesn't do much for that because they're so small, or
You know, I would say, I don't even know if N95s keep mold spores out. Probably a significant portion of them, but definitely not all of them. I don't know what level of protection that they would need. Yeah, I'm not sure. Good question. Okay, so we will, start there when we left off. Okay, so, let's go do our note card for today, and then we're gonna take a break, and then we are going to come back in this room, unless you guys wanna go to the lab. Does anybody care here versus the lab?
No, this is right.
This is better, okay. Virus, where are we? We are here. So we have 2 note cards today. If you have, go grab 2. There's some extra in the back. So this is penicillium, and SP means several species. So SP, that's just the abbreviation. I don't know why it's SP, but it's several species are of penicillium. So the genus is penicillium, the species, there's all different kinds. What do I need to do? I want to go down and grab some pipettes. Is there anything you can think of that we would need from the lab?
I was just gonna grab some pipettes.
Yeah, because we got rid of all of our index stuff too, right? So we're done with everything. They could just finish their lab. Okay.
Do people say they look like little handprints?
Usually what penicillium is described as is paintbrushes. Yeah. So they look a little bit different than aspergillus, but what kind of spores are these? Are these conidiospores or sporangiospores?
Rangia.
So these are conidiospores because the spores are open
Oh.
to the environment, where sporangiospores, remember that has spore and it has 2 spore words. So the spores are inside another whole spore.
You said these were sporangia?
Those are conidia.
Oh, Canada, okay, I was like, wait a minute.
Yeah, now I'm confused. Okay, here's penicillium. You ready for the back? Okay, so it is a mold or filamentous. Don't forget those mean the same thing. Opportunistic. Now we really know that, right? They have a small vesicle. The only reason I put that on there is because it's different than the is it, what did I want to say? Oh, let me wait, I'll show you a picture. Oh no, that's not right. These are not sporangiospores. Sorry, that was a late nighter. These are conidiospores. Not even 100% sure that's how you spell it. Is it conidiaspores or conidiospores? Conidio, okay. So the back of this one's easy, right? But the reason I wanted to bring it up is because I wanted you to know what it looks like. We've already talked about it. They can cause illness. So the reason I put all of that in gray is because they're not highly infectious. People that get a penicillium infection, it's pretty rare. And if they have a penicillium infection, it means they're immunocompromised. But there are penicillium spores everywhere. That's how Alexander Fleming found it, is because a spore landed on his bacterial agar plate and caused a clearing because that mold grew, and that mold grew, it created the beta-lactam and killed off the bacteria around it.
How did you decide that it was?
So he just looked at it under the microscope. So under the microscope, he found that.
Gotcha.
Yeah. Okay, so it can be respiratory. I'll talk more about invasive next week, but when there are, so for example, like you have vegetative hyphae and aerial hyphae, right? Vegetative hyphae grows down. So if your patient has vegetative hyphae and they're growing down further into like the dermal layers, for example, if those hyphae reach the bloodstream, then what can happen, remember that hyphae or spores or anything can get into the bloodstream, and then once you have it in the bloodstream and it can find a little area with enough oxygen or enough moisture or something to live, then it can cause little tiny like micro infections everywhere. So it's really rare. Fungal sepsis is not very common. Fungal sepsis usually happens with severely immunocompromised patients, but just so you know, patients can get invasive fungal infections where it gets into the bloodstream. Okay, so this is what penicillium looks like. They can be all different colors. This is more typical. They're more like this greener color, but they can be different colors, mostly depending on temperature. Temperature changes fungal infections and fungal organisms a lot. So these conidiospores can get huge. They can get really, really long.
Why on Alexander Fleming's agar plate, like it was clear, it didn't look like anything was really growing on it. Why wouldn't it make that kind of structure?
Like, why wouldn't it make this structure?
No, so like you go back to like that, like why didn't it, not that, going back, why didn't it make those structures on this?
Oh, because it was still tiny.
Oh.
Yeah, but if you let it grow and grow,
So that's the first
yeah,
environment,
yes,
but-
yeah, it was just, it had just started an infection,
Okay.
like propagation growth. Okay. Yeah, so on some of our plates, we might not even see that there is a fungal contamination on some of our plates, because they grow a lot, and they grow kind of everywhere. And really, you know, like bacteria can come from our nose and our mouth, but as soon as you open that plate, spores are falling on your plate. So generally, when you see a contaminated plate, sometimes it is bacterial, but most of the time it's fungal. So I'll show you some examples of like some plates we have to throw away because fungal infections grow on our plate sometimes. Yeah.
So we were looking at those photos of the really big glossy things that look really homogeneous and talking about how fungi are like larger than bacteria.
Yes.
Is that like a cluster of fungi that become a homogeneous thing so it looks like just one particle?
Fantastic question. So on those plates that we were looking at earlier that had those big, giant, creamy looking colonies, that's from yeast. So that's what yeast look like when you plate them. And on agar, it'll look like a fuzzy area. This is a mold. So on a mold, on a plate, if you look really, really closely and you kind of look on level with the agar plate, you'll see it looks like fur, you know, just like it does on your raspberries or whatever. So that's what a mold looks like versus a yeast looks like on a colony.
So the yeast would always be like bigger than.
The yeast, they grow a lot and they look like a big, it looks like a giant bacterial colony, where molds, it looks like fur.
Okay.
Yeah, awesome. Okay, so here's the difference between Aspergillus and Penicillium. So Aspergillus is the one that causes black mold. So the reason I put small vesicle here, because under Aspergillus, I put large vesicle. So that's how you can tell the difference between Penicillium and Aspergillus. It almost looks like they have a sporangiospore, but you can tell it's not a sporangiospore because it has spores on the outside. So it's just this big pocket, versus these ones look more like paintbrushes. These ones look more like a lady with funny hair. Yeah? Maybe? I just made that up. Does that help what?
That looks like
A peacock feather?
Yeah.
Yeah, this looks more like peacock feathers versus a paintbrush. Awesome. Okay, so here's Aspergillus. This is our second note card. Several species of Aspergillus can cause infection. Aspergillus is more virulent than Penicillium, and it's more common. It's a more common fungal infection than Penicillium. Aspergillus, we worry about this one a lot because they do create a lot of mycotoxins. So everybody have like the vesicle shape and the chlamydiospores. Why I put sporangiospores again? Don't work late at night. It's not helpful. Okay, so mildew a lot of times will be Aspergillus as well. So not every Aspergillus species is highly virulent. There are a lot of species of Aspergillus that just grow around and grow things. And even black mold wouldn't be that bad if we weren't if people didn't get chronic exposure like living in their homes. Or if it's growing on food and there's the food, even if it's growing, it is growing on food. Aspergillus is everywhere, even right now, at every given time. But the reason that it doesn't affect us is because it's not a huge growth of Aspergillus and we didn't concentrate it into peanut butter. So we worry about things like an Aspergillus infection if it's a large infection. So they're mold, filamentous fungi, opportunistic, large vesicles, conidiospores, mildew, black mold. This is why clothes smell when you leave them in the washer too long, which I do every single time. Respiratory, so sinusitis, so a sinus infection, pneumonia. It can be invasive in the bloodstream. It can cause liver cancers because of the mycotoxins. And then we actually use Aspergillus to make sake, tempeh, and sometimes citric acid. So we can use fungi for all kinds of different things that are beneficial as well, depending on the species. 180 different species of Aspergillus.
And is mold a good, or is bleach a good way to kill it?
Yes, bleach is a great way to kill mold, yeah.
I've heard that bleach doesn't,
Yeah, I can imagine because of the spores that it's really hard to get rid. And yeah, it is really hard to get rid of. But yeah, I think bleach is what's used for the most part. It would help to dry things because they do like things moist. But for example, like my sister that lives in San Diego can't keep bread on the counter, even overnight. Overnight, her entire loaf of bread is covered in mold in one night because it's so moist there. So like, would you be able to get rid of something? You can't keep things that dry. Like she can't, if she keeps her clothes in the wash machine for more than 20 minutes after the washer is done without putting them in the dryer. Like I hang up a lot of my clothes to dry, and she can't because it will grow. It just is so humid. Yeah. Okay. So here's what Aspergillus looks. It looks like a lot like Penicillium. Aspergillus niger is the bad one. Aspergillus niger is black mold. But there's lots of different Aspergillus species. Yeah. Oh, this is one thing I wanted to tell you that was really cool. So this was called the curse of the pharaohs. So there's been this whole thing of like, oh, this is the curse of the pharaohs for thousands of years, that grave robbers would go into mummies' tombs and steal all their gold and jewelry and everything like that they were buried with, right? And then the grave robbers would get super sick and we'd be like, oh, it's because they're cursed because they were grave robbing. Well, actually they figured out that it was Aspergillus growing on the dead decomposing material, and they would inhale so much of those spores, so many of those spores from the mummy, that they would get sick. So I don't know, I just thought that was kind of a cool story. Okay, that is it. Okay, so what I want you guys to do is, we're going to take a break. And we're going to take kind of a long break because I want you to get done with your post lab. So take a break, let your brain have a break, walk around, grab some water, come back, finish your post lab, and then we'll do a review and be done. So let's do, how long do you guys need to finish your post lab? You guys tell me. How many?
I think there's like,
For the first part of the second.
yeah.
2 for the
7
first, 5
questions.
for the second. So do you guys need
Probably like 15 minutes.
15 minutes plus, so what about 3:30 and then we'll do a 3:30 to 4:30 review? Or would you guys rather just get it done, get it going? You guys pick.
Great.
Okay, so let's do a break.
Break for sure.
Let's come back at, we'll reassess at 3:15. Let's reassess at 3:15. If you guys are ready to go, your post labs are done, you're ready to start going, we'll go at 3:15. If you guys still need more time, still need more break, we can wait till 3:30.
Can you flip back to the
Yes.
this?
Yeah, of course.